According to an article written by lead author Emily Mayer, there may be a novel treatment against cystic fibrosis. The article, Quorum Sensing Downregulation Counteracts the Negative Impact of Pseudomonas aeruginosa on CFTR Channel Expression, Function, and Rescue in Human Airway Epithelial Cells, published in Frontiers in Cellular and Infection Microbiology, explores the use of molecules designed to inhibit interbacterial communication during gene therapy. The aim of the study was to compare the effect of the wild-type, engineered and natural mutants and clinical isolates of P. aeruginosa to determine the role of LASR quorum sensing in cystic fibrosis transmembrane conductance regulator, or CFTR, impairment. The second aim was to test the efficacy of a quorum sensing inhibitor, or QSI, against the effect of P. aeruginosa quorum sensing on the wild-type CFTR and on the correction of the delta F508 CFTR mutation by corrector molecules. QSIs have reportedly reduced the bacterial production of residues harmful to the host and increases the susceptibility of the bacteria to current drugs such as Cladico and Orkambi. P. aeruginosa is a gram-negative bacterium that impairs the normal functioning of CFTR proteins in airway epithelial cells, or AEC, and has reportedly interfered with treatments designed to correct this mutation in patients. It readily adapts to its host organisms. Isolates from chronically infected CF sufferers exhibit host-adapted genotypic and phenotypic mutations, for example in the LASR genes. LASR genes are the primarily regulating genes for quorum sensing. P. aeruginosa negatively affects CFTR functional rescue. Exposure to the exoproducts of two strains of P. aeruginosa reduced the maturation of delta F508 CFTRs and inhibited the ability of molecules responsible for correcting the mutation. This study produced results to show that QSIs were responsible for the mutations causing function loss in LASR and virulence genes. Normal CFTR channels allowed for chloride ion efflux from the AEC, which created an osmotic imbalance to draw the water out of cells to moisten the mucus lining. The movement of water into the mucus is to ensure that the mucus does not become thick enough to prevent normal cellular function and movement. However, the water flow out of the cells is not so much that the pathogen trapping ability is neutralized. The expelling of inhaled pathogens is done by the beating action of the cilia. In particular, the study has looked at the delta F508 mutation. This mutation is classed as the second most severe of 5 levels of cystic fibrosis. It is also the most common mutation in Australia. The CFTR, as a result of this mutation, fails to mature and is degraded by proteases in the endoplasmic reticulum. In cystic fibrosis, the CFTR gene is mutated in some way. For example, the phenylalanine at position 508 in the polypeptide chain may be deleted, leading to a change in structure and therefore a change in function, resulting in the delta F508 CFTR mutation. A faulty CFTR channel does not permit chloride ion efflux. No osmotic imbalance can occur, so the water cannot leave either. The mucus becomes very viscous as a result and must often be removed by regularly abusing the patient's back and chest. Unlike normal mucus, this thick mucus does not allow the cilia to beat properly, so trapped pathogens remain in the mucus and are allowed to accumulate on the AECs. This is why cystic fibrosis sufferers experience recurrent infections. The incidence of cystic fibrosis is 1 in 2,500 Caucasians in Australia, with 1 in 25 people being carriers for this homozygous recessive genetic disease. Their life expectancy is approximately 38 years. Fortunately, while no cure has yet been found, the use of QSIs may lead to novel treatment plans. Quorum sensing is the communication between bacteria using small diffusible signaling molecules that activate expression of genes that control multiple factors, including virulence. Keeping this in mind, inhibiting quorum sensing with molecules such as franiol, or HDMF, would inhibit communication and gene expression between bacteria. Inhibiting quorum sensing and therefore virulence production had the added benefit of abrogating the negative effect of P. aeruginosa on the wild-type CFTR, as well as preserving the efficacy of CFTR correctors despite infection. There are other studies that suggest using franiol lower than the non-inhibitory concentration for P. aeruginosa excludes selective pressure for resistance development in the bacteria. AHL signaling molecules are produced at low concentrations by each bacteria in the biofilm and diffuse out into the environment. Once a certain threshold concentration of AHL is reached, the AHL signals interact with cognate receptors to activate transcription of virulence genes. Because the genes that code for signal production are among these target genes, this creates a positive feedback loop. Quorum sensing inhibitors, such as franiol, can either inhibit the production, transmission, or reception of the signaling molecules. S-adenosyl methionine, or SAM, and its analogues are able to achieve signal synthesis inhibition, though it is currently unknown how. Preventing transmission of the signaling molecule can be achieved through alkaline hydrolysis at a high pH or through the use of bacterial enzymes isolated from environmental samples. Reception of the signaling molecule is inhibited through competitive and non-competitive inhibition by AHL analogues and molecules that target the LuxR-type receptor respectively. These pathways may provide novel treatments for cystic fibrosis sufferers and provides an alternative method of treatment from lung transplantations. Future research based on this study may significantly increase the life expectancy from 38 years, possibly to the life expectancy of the average human, which is currently twice more than that. Future prospects of quorum sensing inhibition include avenues in assisting in lung tissue repair and as adjuvants for the increased efficacy of current antibiotics against a myriad of antibiotic-resistant strains currently thriving in the environment.